So, greetings to all of you. This is Teacher Dr. Mugomba Ronald. We are continuing with our series about metabolism or respiration. And in this video, we are going to be looking at the tricarboxylic cycle, what is commonly known as the Krebs cycle. That is what we are going to basically be looking at. Previously, we looked at the process of glycolysis. We saw how the process takes place involving splitting of glucose to produce energy into two pyruvic acid molecules. So we are going to pick on from there. This is Teacher Dr. Mugomba Ronald. Consider to like, share, subscribe, comment down in the comment section. Krebs cycle or tricarboxylic acid cycle. It is known as tricarboxylic acid cycle because when you look at most of the molecules that we are going to be looking at, the intermediates in the reaction, are having 3 carboxylic acid groups. And it was discovered by a man known as Krebs. So that is why sometimes it is called Krebs cycle. So when you look at the Krebs cycle, it's the final pathway where oxidative metabolism of carbohydrates, amino acids, and the fatty acids converge, their carbon skeletons being converted to carbon dioxide. This cycle occurs totally in the mitochondria and is therefore close proximity to the reaction in the electron transport chain. Unlike glycolysis, which happens in the cytoplasm, this one purely occurs in the mitochondria, and anything outside the mitochondria does not take place, and it only takes place in aerobic conditions or in anaerobic conditions in the presence of oxygen. That's when it will take place, because oxygen is required as the final electron acceptor. So the whole process begins with a simple process known as oxidative decarboxylation of pyruvate. Pyruvate, a 3-carbon molecule, first undergoes a process known as oxidative decarboxylation. During oxidative decarboxylation, the end product of aerobic glycolysis must be transported to the mitochondria before it goes into the tricarboxylic cycle. This is usually accompanied by a complex of enzymes. So we are going to go ahead and look at the oxidative decarboxylation. So what usually happens is, when you look at this process known as oxidative decarboxylation, we have our pyruvate here. This pyruvate undergoes oxidative decarboxylation. Oxidative decarboxylation in simple terms is we are carrying out oxidation with emission of carbon dioxide. So it is called decarboxylation simply because of that fact. When you look at it, you can see that this oxidized NAD comes into the cycle and leaves as it is reduced. So this is enough to tell us that the process is an oxidation process, because the hydrogen ions that are released, or the protons, they are immediately accepted by the oxidized NAD to become reduced NAD. But in the due course, you can clearly see that we can see that a coenzyme A is also being brought in. So as we are losing a carbon atom from pyruvate, we can see that a coenzyme is brought in. So we can see a coenzyme being brought in. So the coenzyme then combines with an acetyl group produced from the decarboxylation of pyruvate. So we end up with a molecule known as acetyl coenzyme A. So you can see from here, pyruvate plus coenzyme plus oxidized NAD produces carbon dioxide, acetyl coenzyme A, and reduced NAD. You can clearly see that. So that is the process of oxidative decarboxylation. It prepares this molecule so that it can be received by the enzymes which are found in the mitochondria. So that is oxidative decarboxylation. We move from pyruvate to acetyl coenzyme A. How? By getting rid of carbon dioxide. We get rid of the carbon dioxide, and then after getting rid of the carbon dioxide, we can see that this happens. This is a transition state. Sometimes they are called a transition state. So we add in a coenzyme, get rid of the carbon dioxide, and we form acetyl coenzyme A, which is a 2-carbon molecule, which is going to go ahead into the Krebs cycle and then it participates in the different reaction. It comes as a reactant in the Krebs cycle. So you can clearly see that reaction that we have just been talking about. You can clearly see that reaction which we have been talking about, oxidative decarboxylation. It happens in the presence of an enzyme known as pyruvate dehydrogenase. So you can see we have moved from during oxidative decarboxylation, we move from a 3-carbon molecule to a 2-carbon molecule as enzyme A. You can clearly see that. Now, I will first talk about a number of things. Now, when you look at this whole process when you look at this whole cycle, it looks a little complicated to you. But I'm going to go ahead and give you an acronym to remember all of them. But you can start by filling in the boxes. You can do the cycle and then you start filling in. To start with, you can see the first one is called citrate, the second one is isocitrate, the third alpha ketoglutarate, then succinyl CoA, then succinate, fumarate, malate, and then oxaloacetate. Now, you can remember with this simple mnemonic, citrate is Krebs starting substrate for making oxaloacetate. You can remember it. Citrate is Krebs starting substrate for making oxaloacetate. Citrate is Krebs starting substrate for making oxaloacetate. It's an acronym that will help you to remember, a simple mnemonic that will help you to remember these terms. So citrate is standing for citrate. Is stands for isocitrate. Krebs stands for alpha ketoglutarate. Starting stands for succinyl CoA. Substrate stands for succinate. 4 stands for fumarate. Making stands for malate. Oxaloacetate stands for oxaloacetate. Now, let us go ahead. Now, another way we can remember, I would like to give you another mnemonic to remember the enzymes which are involved in each of the stages. 5-dede, 5-dede, 5-di, maro di, 5-dede, 5-di, maro di. That is the acronym that we can use to remember the enzymes. Now, when you look at the whole process, it all begins from here. You can see that enzyme A comes in. So you can see enzyme A comes in here, and it binds. This enzyme A is a 2-carbon molecule. It reacts with a molecule known as oxaloacetate, which is a 4-carbon molecule. You can see it clearly that it begins with enzyme A binding with a 4-carbon molecule known as oxaloacetate in the presence of an enzyme known as citrate synthase. So in the presence of citrate synthase, we end up having a molecule known as citrate. So the enzyme is known as citrate synthase because it catalyzes synthesis of citrate. Now, after there, we go ahead and see our next reaction here is we are converting this citrate into isocitrate. And the conversion of citrate into isocitrate is known as, we can see the enzyme is aconitase. Aconitase is the enzyme that catalyzes the conversion of citrate into isocitrate. So isocitrate is like an isomer of citrate. It is still a 6-carbon molecule, the way you can see it on the screen. It is 6-carbon. And now from there, what happens thereafter is we are going to undergo dehydrogenation reaction, removal of a proton from isocitrate. So when we are removing the proton, the electron acceptor that is going to take away the proton is NAD. Oxidized NAD will come into the reaction and it will leave as reduced NAD. In that way, we are seeing dehydrogenation of isocitrate. Dehydrogenation or oxidation of isocitrate. Then in the due course, but remember, when we were coming into this reaction, we had 2 enzyme A. Anyway, we shall talk about that computation. So that happens in the presence of isocitrate dehydrogenase. So the enzyme is isocitrate dehydrogenase because it is carrying out dehydrogenation of isocitrate. And the electron acceptor that is here is NAD. It is the one that is receiving the protons. Then after there, it converts to what we call alpha ketoglutarate. Now you can see clearly that this one is called alpha ketoglutarate. Now alpha ketoglutarate further undergoes a process known as dehydrogenation. Dehydrogenation and at the same time decarboxylation. That is why you can see that we are getting rid of protons which are being received immediately by oxidized NAD to form reduced NAD. But at the same time, we can see that we are also producing a carbon dioxide molecule. You can see that carbon dioxide molecule that is here. You can clearly see that carbon dioxide molecule in this area here. So it is both the dehydrogenation process and the decarboxylation process. This explains why succinyl coenzyme A is it's a 5 carbon molecule. It is a 5 carbon molecule, not 6 carbon as the other molecules, because we are seeing loss of a carbon atom. Now that takes us to, in fact it is a 4 carbon. It is a 4 carbon. Isocitrate is alpha ketoglutarate is a 5 carbon molecule. When you look at isocitrate, it was a 6 carbon molecule. Isocitrate was a 6 carbon molecule, but during the conversion of isocitrate to alpha ketoglutarate, we also see some release of carbon dioxide. So isocitrate to alpha ketoglutarate, you can see clearly that we are moving from 6 carbon, where isocitrate was a 6 carbon molecule. It loses a carbon dioxide molecule as a result of decarboxylation and dehydrogenation at the same time. So here we also have some carbon dioxide being lost in this area here. So because we also see some loss of carbon dioxide, that's how we have alpha ketoglutarate being a 5 carbon, and then more loss of a carbon. Then you can see succinyl coenzyme A is also a 4 carbon, because we are seeing another release of a carbon. Now thereafter, you can clearly see that reaction which involves alpha ketoglutarate being converted to succinyl coenzyme A is a dehydrogenation reaction. That is why we are having alpha ketoglutarate dehydrogenase enzyme in place. Now we have our 4 carbon molecule here. You can clearly see that it is a 4 carbon molecule. Now when we are converting this succinyl coenzyme A to succinate, it involves an enzyme known as succinyl coenzyme synthase. Because Because we are converting succinyl CoA into succinate. So it is an enzyme known as succinyl coenzyme synthase. Good. So when you look at this reaction here, it involves expenditure of some energy. This energy that is expended is immediately taken up by GDP. But this GDP and an inorganic phosphate, they turn to GTP, and this GTP is immediately converted to this one. GTP usually is immediately broken down. This energy that is kept in GTP is immediately taken up by ADP and an inorganic phosphate to form ATP molecule. So that is why some books you find this, it is directly forming ATP instead of GTP, because the energy that is immediately taken up by GTP is immediately stolen from it by ADP and an inorganic phosphate to form ATP. So we see here a reaction which involves direct substrate level phosphorylation. Substrate level phosphorylation is we are seeing production of ATP at substrate level. We are seeing, as we are converting one molecule to another molecule, we are seeing ATP being converted. That is one of the ways we can produce ATP. Now, in that reaction where we see succinyl CoA to succinate under the catalysis of an enzyme known as succinyl coenzyme synthase. Now, from there, that takes us to succinate. Now, this succinate is usually converted into fumarate. Now, this conversion of succinate to fumarate under the presence of an enzyme known as succinate dehydrogenase. These protons that are released from the dehydrogenation of succinate to fumarate are immediately taken up by FAD. Here we are representing it as Q. Q, it becomes reduced to Q QH2. In books, some books you'll find it being called FAD, being reduced to sorry about that. So we usually see FAD being converted to reduced FAD or FAD H2. So this one here, succinate to fumarate, releases protons which are used for synthesis of FAD. Now, from fumarate to malate, it involves a hydration reaction where we can see in the presence of an enzyme known as fumarase, we see splitting. That is why they call it, we see addition of water, as you can see it here. On top of addition of water, we are seeing formation of malate. That is the reaction. So it is a hydration reaction. So we end up with malate. Now this malate here is going to go ahead and becomes dehydrogenated to oxaloacetate. So the dehydrogenation reaction is catalyzed by malate dehydrogenase. These protons that are produced here, they are going to be taken up by a proton acceptor known as NAD, and then NAD is converted to FAD. Sorry, NAD is converted to reduced NAD. In the due course, we form oxaloacetate. So you can clearly see in this reaction here, if we are to look at it carefully, remember I told you from the start that this reaction here involves, we had 2 molecules of pyruvate. That means if we had 2 molecules of pyruvate, as we are converting 2 molecules of pyruvate to acetyl coenzyme A, we also form the 2 acetyl coenzyme A molecules. So if we have 2 acetyl coenzyme 2 molecules, that means we form the 2 citrate molecules, and that means that we form the 2 isocitrate, and that means that we also form the 2 reduced NAD in a reaction of isocitrate to alpha ketoglutarate. Now that means that we also formed another 2 at alpha ketoglutarate to succinyl CoA, and then that means that even here when we are seeing succinyl to succinate, we also formed 2 ATP molecules. This means that even when we are converting succinate to fumarate, we also produced 2 FAD molecules, which here we are calling Q. That means that even here where we are converting malate to oxaloacetate, we produced 2 reduced NAD. That is what it means. Now let us go ahead and see about the figures. So this means that from this reaction we have formed a total of 6 reduced NAD. We have also formed a total of 2 reduced FAD, and we have formed a total of 2 ATP molecules from this reaction. So I think you can see that computation that is here. In glycolysis, we formed a total of 2 ATP molecules. In the mitochondria glycolysis, still, that is the transition state. We saw that transition. So the glycolysis also produced 2 reduced NAD, and those reduced NAD, when they are taken to the electron transport chain, they will produce 6 ATP molecules. And then when you look at the aerobic respiration like that takes place, now we have even the transition state produced, this transition state here produced 2 FAD, it produced 2 NAD, and each of them, remember, is 3. That gives us 6 ATP molecules. When you look at the Krebs cycle, it formed 3 FAD NAD, but remember we had times 2, so that gives us 6 reduced NAD, and that gives us a total of 24 ATP molecules. When you look at FAD, we had 1 being produced, and we also because each of them gives us 2, that gives us a total of 4. So when you add FAD NAD, which was 6, each giving you 3, that is 18. That is 20. Sorry, that is 18. Now when you add 18 plus the 2 that are produced, the 4 that are produced from FAD, that gives you a total of 22. Now when you also add these ATP molecules, remember we formed the ATP. You remember the reaction which involves succinyl CoA to succinate, that produced ATP directly. So since there were 2 molecules, we had also 2 ATP molecules. That's why we have 24 ATP molecules from the Krebs cycle. So when you make the mathematics, you end up with a 38 ATP molecules. This is theoretical. Most of the times, it is not 38. It is less than 38 practically. So thank you so much for listening. Today we have been looking at the Krebs cycle. We have been looking at how energy is produced from the Krebs cycle. We can see that we have direct production of energy through substrate level phosphorylation. We also see indirect production of energy through protons being used to form reduced NAD and reduced FAD, which are taken to the ETS, where we see oxidative metabolism taking place, and we form the ATP molecules. I remain teacher Dr. Mugomba Ronald, MBCHB. Greetings to all of you. Thank you so much for being around. Consider to subscribe, like, share, comment in case you find it useful to you. Teacher Dr. Mugomba Ronald.